So neuroanatomical techniques, right? Getting inside this structure, right? And figuring out like what are various constitutive components of our neuroanatomy. And actually I was noticing we've dropped this one, so you may actually hear something rattling around inside of it. Neuroanatomy is the study of nervous system architecture. And we can talk about sort of gross structures. And that actually means like large scale structures. So, lobes of the brain, like the frontal lobe or the occipital lobe, right? Or the cerebellum, for example. And that's in contrast to, aspects of kind of fine neuroanatomy, like, the dendrites or the axon terminals or the myelin sheaths, right? Or, individual nicotinic cholinergic receptors that we have, localized sort of presynaptically at axon terminals. So in order to identify, aspects of fine neuroanatomy and also to see more deeply within, the larger scale structures, a couple of developments had to take place. For one, we had to develop what are called sort of fixatives, things that could fix the brain, make it firmer and stronger and kind of more, steady, so that we could then use a kind of a slicer. Think like a deli slicer. It's called a microtome. So it can make very, very thin slices of tissue. So the fixatives, and for outreach we use something called formaldehyde, right, which helps to or formalin, which helps to fix the brain and make it sturdier so that it can be sliced, for example, and it will last longer. And then, of course, it required the development of microscopes, in order to see, smaller scale structures, the fine neuroanatomy of the brain. There's the electron microscope, which can resolve very, very small, aspects of fine neuroanatomy. And I've got some images up here, for example, of like a nicotinic cholinergic receptor. And it's just remarkable. Other techniques like X-ray crystallography, which can, show you the actual physical structure of these individual protein machines. Really extraordinary advances in technology that have allowed us to get much closer in at the fine neuroanatomy of the brain. So one of the major advances, actually, in the study of neuroanatomy, particularly fine neuroanatomy of individual neurons, was the development of something called the Golgi Stain. And this is initially referred to as the black reaction. But Camillo Golgi, who was an Italian neuroanatomist, who won the Nobel Prize along with Ramon Cajal, who drew some absolutely astonishing images, of neurons from the human brain utilizing Golgi's black reaction or the Golgi stain, for the recognition of their work on the structure of the nervous system. It really allowed, scientists to see, all the various components of a neuron. So this silver staining process would actually, blacken, all the various compartments of a single neuron. Including the dendrites, the dendritic spines, the soma, the axon hillock, the axon collaterals, and the axon terminals. So it really revealed the entire, structure of an individual neuron. One of the things that's extraordinary, and Golgi was also an artist, an accomplished artist, Italian neuroanatomist and artist, who drew, a beautiful image, a famous image of his of the hippocampus and the circuitry in the hippocampus. What's interesting is that this black reaction only stains about 1% of the neurons that are found in a slice of brain tissue. And that's pretty remarkable, because when you look at it, it looks like there's a lot of neurons in those images to some extent, but that's only 1% of what's there. So really the actual, brain, if you were to stain everything with the Golgi stain, it would be just a black sheet. You wouldn't see anything at all. So Golgi staining, also called the black reaction, was first discovered apparently by accident by Camillo Golgi, and it could selectively visualize the entire architecture of a neuron, with a clear background that thousands of neurons next to it were left, basically unstained, making it possible to investigate the neuronal morphology, under a microscope. The architecture of the impregnated neurons, including cell somas, axons, dendrites, and spines, could be easily visualized.